Hello, I'm glad you're here. If you're watching this video, you likely already know who I am. I should have sent you an invitation. I doubt that the YouTube algorithm brought you here, so I don't need to introduce myself. Now, what's this video about? Well, I've had some things on my mind for a long time that I want to discuss, and this seemed like a way to do that. Or maybe I bought a new camera and I need to justify the amount of money I spent on it. Or maybe I bought a new computer and I need to justify the amount of money I spent on that. I guess you can be the judge. In making this video, I originally wanted to speak extemporaneously from an outline, but I just got too tongue-tied and gave up. So I wrote a script and I'm using my teleprompter. I tried getting AI to make the presentation for me, but it was too creepy. So here I am, the real me. But what I want to do is to touch on some of the high points of modern philosophy and the philosophy of science, and I then want to apply them to what we discern from evidence in modern molecular biology. If you studied modern biology, my discussion of biochemistry will be just a high-level overview. On the other hand, if you're a baby boomer and your knowledge of biology dates back to the 1960s and 70s, this may be something of a deep dive. Now, you likely know that in anticipation of retirement from my engineering career, I studied at WSU to get my teaching certificate with an endorsement to teach physics. But WSU required that, against my will, I would study biology and get updated. So I took a 3-credit course in modern biology from the University of Missouri, and let me tell you, it just blew me away. I would go as far as saying a love for biology largely displaced my fascination with physics, and I've been fascinated with biology ever since. I had no idea about what had changed since Mr. Rose's 10th grade biology class in 1964, nor how amazing it was. So I want to talk about what we now know about molecular biology. And spoiler alert, I'll want to consider whether the Darwin of the gaps is able to rescue evolutionary theory from its multiple challenges. In the world of science, scientists investigate things, collect information, and then infer to the best explanation of what they found. In the 14th century, the Franciscan friar William of Ockham said that we should seek the simplest explanation to explain what we observe. Fast forward 300 years, and we have the Enlightenment. Two consequences of the Enlightenment were the propagation of rationalism and the propagation of materialism in modern philosophy. In rationalism, we rely only on our intellects to discern truth, while materialism demands that there be an explanation for everything we see, touch, or otherwise discern based solely on the laws and constants of nature. That doesn't mean that we already have explanations for everything or that we ever will have the explanations for everything. But ultimate explanations will nevertheless always flow from natural laws and constants. This, of course, precludes the intervention of any divinity. For some, this precludes the entire concept of a divinity. In the 20th century, two interesting philosophers of science emerged, Karl Popper and Thomas Kuhn. Popper is known for his analysis of the international order that evolved after World War II and the potential use of nuclear weapons, but also for his ideas about how a scientific theory might be found to have explanatory power. To have explanatory power, you must be able to apply one or more tests to a theory that have the potential to show the theory false. The theory must be falsifiable. For example, Albert Einstein's general theory of relativity predicts the existence of gravity waves. If appropriate tests fail to find these gravity waves, then the theory might be stripped of its explanatory power. But here in Richland, the LIGO Observatory found these gravity waves. This led to the award of a Nobel Prize. So the result of this investigation supports a conclusion that the general theory has explanatory power. Note that we don't say it proved the theory, only that it showed it had explanatory power. Popper would note that we are always testing theories in a regular advancement of our understanding and confidence in the theories. But Thomas Kuhn wasn't quite so sure. Are we really always testing theories? Are we really always open to the significance of new information regarding a phenomenon? Kuhn noted that science moves from one paradigm to another, and the road from one paradigm to another might be bumpy. A crisis appears just before the transition from a dying paradigm to a new one. For example, Newton's laws, including his theory of gravity, constituted a prevailing paradigm for hundreds of years. They explained a lot of what we observed about the natural world and satisfied our intuitions. But toward the end of the 19th century, astronomers noticed some problems with the orbit of Mercury. And then, in 1887, the Michelson-Morley experiment successfully falsified Galileo's and Newton's theories of relativity. This was a crisis. After the experimental results were revealed, physicists twisted themselves into pretzels, trying to reconcile the experimental findings with Newton's paradigm. It wasn't until 1906, when Albert Einstein published his famous papers, that it became clear a new paradigm had emerged. But it was only a few years later that the need for a paradigm of quantum mechanics showed up, and poor Einstein was playing catch-up ball. Kuhn noted how hard it was, as with Einstein, to leave an outdated paradigm behind. He also noted that we may not be doing the constant open-minded testing of theories that Popper postulated. Especially since Darwin popularized his theory of evolution, there has been a lot of consternation over a conflict between science and religion. In the past, these had been seen to share a domain. For example, in the General Scholium to his Principia Mathematica, Newton attributed gravity to the ongoing activity of God in the universe. But with the advent of Darwin's evolution, this idea of a shared domain changed. One resolution that's been proposed to resolve the apparent conflict is to say that religion has its domain and science has a separate one. But this hasn't kept religious personalities from denigrating science, and a lot of scientists from declaring that science will, probably sooner rather than later, permanently bury religion. Speaking for myself, as both a religious person and as a person with a great interest in science, I have my own resolution. I have confidence that, one way or another, the Bible is, in fact, the Word of God. With respect to science, I look to distinguish the amazing things that scientific investigation has actually found and from the conclusions that many scientists have drawn from their findings. I want to separate a reasonable conclusion based on evidence from an opinion driven by an underlying philosophy. And I think an honest search for tests of falsity are important. In particular, I wonder if an underlying philosophy of materialism might lead a scientist to a conclusion that's not truly supported by the evidence. Now, in this video, I mostly want to talk about molecular biology. I think I'm able to discuss this in an informal video like this because after taking that course at the University of Missouri, I also taught, for a while, a course called Advanced Biology. We used a text that was appropriate for an undergraduate university course, although this was high school. Also, I still have the text I used at the University of Missouri, and I consult it from time to time when questions pop up in my mind, which still happens. I've also read plenty of books by men like Richard Dawkins and Daniel Dennett, so I understand their perspectives. In fact, the day that the news of Dennett's death rolled across my computer screen, a copy of his book, Consciousness Explained, was sitting on the desk right in front of me. Back in the school year of 1964 to 65, when I took high school biology, very little was understood about the cell. In fact, they were still trying to fit all life into an animal or vegetable straitjacket, such that bacteria had to somehow be classified as vegetable. So even today, doctors speak of gut flora, which are not flora at all. Watson and Crick had only recently elucidated the double helix geometry of the DNA molecule, and Crick was figuring out what it actually did. A few years earlier, the Miller-Urey experiment had demonstrated that natural phenomena, especially involving lightning, could form some amino acids. But even then, questions were being raised as to whether their experiment actually replicated conditions that would have existed at the time life was supposed to have begun. Under the microscope, one could see the nucleus of the cell, but scientists weren't quite sure what its function was. Cells could be seen to divide in an amazing phenomenon, but no one knew yet, and as we shall soon see, how truly amazing that process actually is. And then there were those little dots in the cytoplasm that no one really understood. But especially as the technology of the electron microscope advanced, the truly amazing complexity of the cell came more and more into focus. Francis Crick was showing that the arrangement of nucleotides in DNA corresponded to certain amino acids, and in time the little dots would turn out to be little machines and little factories turning out all sorts of material. We call these organelles, from a German word for little organs. And to the cells, they are like organs in our bodies. Now, if you don't know much about DNA and you don't know what an organelle is, hang on. We'll get to that. We want to consider the design of the cell. Those organelles, that machinery, must be protected from a hostile chemical environment. So they are surrounded by a membrane. If you remember that osmosis experiment from high school, you know that stripping the shell from an egg leaves a membrane. The membrane is actually somewhat porous, so water molecules can cross it, sort of like going through a strainer. And if you immerse the egg in water overnight, water will diffuse across the membrane into the egg. That's because the concentration of water outside the membrane is greater than inside, and water migrates down the concentration gradient. It moves from high concentration to low concentration. We call this diffusion. But if you take that fat egg and leave it in corn syrup overnight, well, it shrivels up like a prune. There are no free water molecules in the corn syrup, so the concentration gradient orients in the opposite direction, from inside to outside. Water flows down the concentration gradient and moves out of the egg into the corn syrup. The same thing happens in the cell. There may be nutrients and gases like oxygen outside the cell, and these diffuse into the cell that needs them. But waste, including CO2 gas, builds up and diffuses out. Pretty cool, right? But there's a problem, or maybe several problems. If the nutrient and gas molecules are much bigger than a CO2 molecule, they won't fit through the porous membrane. Also, if the particles are charged, like chlorine and sodium ions from salt, they can't get through. Why can some diffuse through the membrane and others not? Let's take a look at the amazing design of the cell's membrane. What we have is what's called a phospholipid bilayer. You'll recognize phosphorus as an element that belongs to many compounds. But what's a lipid? Lipids are simply fats, and fats don't get along with water. So the phosphate heads of the phospholipid molecules are exposed to the water on both sides, with the lipid tails between. There's no free water between the layers. But there's enough space between the phospholipid molecules to allow smaller molecules to diffuse through, but not if they're charged. So the membrane is dotted with special protein gatekeepers that facilitate the diffusion of some larger and charged nutrients and gases back and forth across the boundary. Now that's great if the diffusion gradient is going the right way. But what if you need to move nutrients and waste against the gradient, uphill? You have other protein gatekeepers that can actually pump materials against the gradient. This pumping takes energy, and this energy is provided by a chemical we refer to as ATP. ATP is the energy currency of the cell, and we'll have more to say about it in a little while. Also, these gatekeeper proteins must recognize whatever it is that they're passing. They need to recognize material that should pass and reject material that should not pass. So they're very specialized and require a very special design to function. Now, I don't know why, but for some reason, the nucleus has its own double membrane, two phospholipid bilayers layered one on the other. We'll see later that some really important stuff needs to pass out of the nucleus, so that membrane has its own characteristics. Now let's talk about deoxyribonucleic acid, or DNA. Everyone knows that double helix, first described by Watson and Crick when I was 4 years old. It tells everything there is to know about you. As Steven Spielberg showed, if you can get your hands on some dinosaur DNA and inject it into frog eggs, you get dinosaurs. How cool is that? But does DNA really tell everything there is to know about you? Does it dictate whether you have 8, 6, 4, or 2 legs? What is it anyway, and how does it work? Well, like the hard disk on your computer, it's a vehicle on which code is recorded and from which information can be read. It can be thought of as a base for code, where binary computer code is a base 2 code, ones and zeros. The DNA molecule has two opposing backbones and rungs connecting them. You can think of the two backbones like a photographic positive and a photographic negative. The positive side contains data that is to be read, while the negative side is used to form a new positive side during replication. But why is the data read? This takes us to what Francis Crick called the central dogma of molecular biology. In stating the central dogma, people usually say DNA makes RNA and RNA makes protein. In other words, RNA gathers information from DNA that is used to construct proteins. Crick actually had a different and more long-winded definition, which I'll summarize by saying that information passes from DNA to protein, but not from protein back to DNA. The information in question is the design of the protein. You can use your house plan to build your house, but your house can't create its own drawings. DNA provides the design for the construction of proteins, but proteins can't create DNA. And, at least in modern times, there are no proteins without a description of the protein in DNA. Each protein molecule must be constructed using information in DNA. That being the case, DNA must be divided into segments we call genes. How does this work? The backbones are composed of what we call nucleotides, special molecules composed of a phosphate and a sugar. Then each nucleotide has attached one of four what we call nucleotide bases: adenine, cytosine, guanine, and thymine. We'll refer to them by their initials, A, C, G, and T. So the bases project out from the nucleotides and bond to another base on the opposing backbone. But they bond according to a strict rule. Adenine will bond only with thymine, and cytosine will bond only with guanine. And so you have the rungs of the double helix. These are connected by what we call hydrogen bonds, and they are relatively easily broken. This makes it easy to unzip the double helix and read the data. What's important here is that these bases appear on the backbone in a very specific order. This order is the symbolic code describing the design of a given protein. And how does that work? Well, the bases appear in groups of three called codons. A given gene is demarked by two codons. A codon we call a start codon, and one we call a stop codon. Codons in between the start and stop codons can be read, and the information can be used elsewhere in the cell. The sequence of codons can be understood to represent from among 20 amino acids that will constitute a protein constructed according to this design. Now, there are more codons than there are amino acids, so some amino acids could be represented by more than one codon. But going back to our more simplistic version of the central dogma, the DNA will be used to make RNA, and the RNA will be used to make proteins, eventually. At this point in our discussion, it would be good to distinguish between prokaryotic life and eukaryotic life. Prokaryotes are bacteria and archaea, while eukaryotes are everything else. Everything else includes yeast, sponges, groundhogs, and you. Interestingly, it was Francis Crick who recognized archaea as an entirely different domain from bacteria. These are sometimes understandably confused with bacteria, but they are very different. Archaea are the organisms that live in extreme conditions, such as in the near boiling water in the Morning Glory pool in Yellowstone. Archaea give the pool its beautiful blue color. While prokaryotes have their DNA distributed throughout their cells, DNA in eukaryotes is located entirely in the nucleus. Prokaryotes don't have a nucleus. We'll get into how cells reproduce in a few minutes, but we first want to see how DNA replicates, because that has to happen first. It's not simple. All of my explanations of biological processes are seriously simplified. I could say much more about each one. Now, each strand of DNA constitutes half a chromosome, or what we call a chromatid, but they are only bunched up in that famous X shape at the beginning of cell division, mitosis. A chromatid is one half of the X and one strand of DNA. Most of the time, chromosomes are distributed as strands of DNA throughout the nucleus in eukaryotes and throughout the cell in prokaryotes. DNA replication requires the work of a bunch of different enzymes. Enzymes are special proteins that catalyze biochemical reactions. Since they are proteins, they are first described in DNA and later constructed. And so, it seems to me, we have here one of many chicken and egg questions. How were proteins first made when you need proteins to make proteins? In any event, there's a specialized enzyme to break the hydrogen bonds forming the rungs and then zip the double helix. The hope is that the cell can find loose nucleotides and bond them to complementary bases on both backbones. This way, the cell can form two new complete DNA strands. But there's a problem. Unzipping a strand of DNA is like getting too long a strand of adhesive tape. It wants to stick to itself and it likes to coil. So there's an enzyme that needs to cap each newly opened base with a special protein that will keep all the bases from finding complementary bases. Then there's an enzyme that keeps the strands from coiling up. Then a builder enzyme finds loose nucleotides in the cytoplasm and places them on the open strands, thus capping them. So when the open DNA strand has an A base, the builder enzyme will remove the protein cap and place a complementary nucleotide with a T base on it. If there's a G base there, the enzyme will place a nucleotide with a C base on it. The new nucleotides bond together to form the new backbones. The new backbones are mirror images of the earlier backbones on which they are forming. One quirk of this is that the DNA strand is opened from one direction, and the new strand must be constructed from the other direction. So as the strand is opened, a builder enzyme needs to rush ahead and start building the strand backward. When it finishes a segment, the builder enzyme rushes ahead to start where the unzipping enzyme has been working. The nucleotides connect with one another, reconstituting the backbones, and we end up with two identical DNA molecules. Again, this process is a lot more complicated than I've described, and it involves many more enzymes. I really struggle to see how such a complex process could have evolved from a simpler process in a stepwise fashion, especially since this is the process that's claimed to produce the changes necessary for evolution to occur. According to evolutionary theory as it currently exists, mutations of genes would have incrementally contributed to the survival of the organism and crowded out the earlier version. That would occur due to natural selection. Would there be intermediate steps leading up to the amazing process, each step contributing a new ability to survive? It's a long way from some nucleotide bases floating around in a soup to nucleic acid molecules coding for specific proteins. A lot of incremental steps. It's during this process of replication that errors can be made, introducing mutations at points on the genome. So we call them point mutations. Current thinking is that these mutations may give an organism an advantage, such that when acted on by natural selection, the organism will survive when it might otherwise have become extinct. Now, after the DNA has been replicated, the cell can move on to the process of division we call mitosis. We'll look at how mitosis occurs in eukaryotic cells, those with nuclei enclosing the chromosomes. The process in prokaryotic cells is a little different, but not much simpler. The first thing to notice about all of this is how so many different processes must be coordinated in time. So it seems that some kind of clock is ticking. Now, mitosis occurs in 4 stages, or 5, or 6. It depends on who you ask. First, you have prophase, when the strands of DNA condense into chromatids and are then bound together into the famous X form of the chromosomes. The strands wind around special proteins called histones. Remember, all of the proteins exist only because they were first described in the DNA and later constructed from these design descriptions. And so the chicken and egg problems continue. The chromosomes consist of 2 identical chromatids, which are bound together at the waist by a structure called a centromere. A centromere is a region of the DNA together with a specialized histone that causes the 2 identical sister chromatids to join. Some see what happens next as a separate phase, and they call it prometaphase. This is when the nuclear membrane breaks up so that it is not a barrier to the succeeding steps. At this point, structures called mitotic spindles form. These migrate to what will become opposite poles of the cell. When they position themselves there, we call them centrioles. That's the end of prophase. Now metaphase begins. In metaphase, fibers shoot out from the centrioles and attach to the centromeres of the chromosomes. Each chromosome will have fibers attached to it from both centrioles. I call these fibers, but they are actually another organelle called a microtubule. In any event, working through the microtubules, the centrioles maneuver the chromosomes to line up along the axis of the cell. After metaphase, we move on to anaphase. In anaphase, the fibers shrink, pulling apart and separating the chromatids. They then pull them to opposite sides of the cell. After anaphase, we have telophase. This reminds me of what our computer does when it installs new software. The installation process leaves behind a mess, and there needs to be a process to clean up the mess. In this case, we need to form new membranes around the chromatids gathered on each end of the cell. These, of course, are the double phospholipid bilayers that we discussed before. Also, the chromosomes decondense into loose strands of DNA available to be read for the information they contain. Now, some will end their description of mitosis here, leaving the actual division of the cell outside of mitosis, while others will include it. But the actual division occurs when a tiny fiber is wrapped around the outside of the cell, halfway between the regions where the new nuclei formed. This fiber then shrinks until it cuts the cell in two, and so ends mitosis. The process of meiosis has similarities with mitosis, but it's much, much more complicated. And if we tried to get into it, this video would be much, much longer. But meiosis is the process by which gametes reproduce. Gametes are both the eggs and sperm cells in sexual reproduction. This process resembles the 4 stages of mitosis, but then goes back and does another 4 similar stages. Now, this process is essential to sexual reproduction, and so it must preserve some traits of both parents, making decisions about which trait will carry forward from which parent. To facilitate this, the gamete contains a full set of chromosomes inherited from each parent. So, in this process, something really weird occurs. There's a sequence of events by which the genes that code for one trait are clipped out of one parent's chromosome and swapped out with the genes that code for that trait in the other parent's chromosome. We call this process crossing over. This is pretty amazing, but it's essential to how sexual reproduction occurs. In one gamete, there may be a swapping of genes for eye color, but in another, there is no swap for eye color. There are lots of gametes, so there is a lot of variety in which genes from which parent will be carried forward. Some years ago, I did read Darwin's The Origin of Species. I'm not sure I finished it, but I was struck by how clearly and cogently it was written. In proposing the process by which evolution occurs, Darwin said that it must proceed in small increments, and each increment must provide a survival advantage to the organism. How did all the processes of sexual reproduction originate, and how did the machinery that facilitates them come to be? This in a stepwise manner, where each increment contributes to the survival of the organism. There are so many independent parts that must work together, and they're all required for sexual reproduction to work at all. How life progressed from asexual reproduction to sexual reproduction is a mystery to me. Those who study this while holding to a materialistic philosophic worldview invoke the Darwin of the gaps. Genetic mutations, acted on by natural selection, have the creative power to do all of this. We don't yet know how, but someday maybe we will. But to me, this seems a bridge too far. Does the Darwin of the gaps really have the creative power necessary to overcome the many chicken and egg problems? Can it build new systems of reproduction, energy processing, protein synthesis step by step? You can, of course, draw your own conclusions, but I sure don't see it. Now, I want to return our attention to the central dogma of molecular biology. Remember that? Or perhaps we might refer to protein synthesis. Information about the design of protein is encoded in segments of DNA we call genes. The information must be transcribed from the DNA and translated into a protein. How does that work? Well, it's not simple, but it's really amazing. First, recall that like computer code, information is recorded on the DNA strand. It is, however, not a base 2 code with ones and zeros, but it can be thought of as a base 4 code with the symbolic letters of A, C, G, and T. These letters stand for the nucleotide bases adenine, cytosine, guanine, and thymine. A cannot bond with C, G, or itself, but only with T. C and G can only bond with each other. These bonds connect the two backbones of the double helix, but the bonds can be broken by an enzyme to unzip the DNA molecule. The intermediary in the process between transcription and translation is another nucleic acid you've surely heard of before, RNA, or actually three of them, transfer RNA, or tRNA, messenger RNA, or mRNA, and ribosomal RNA, or rRNA. Like DNA, RNA is a nucleic acid composed of a string of nucleotides. They're the same four as in DNA, except it uses a base called uracil instead of thymine. And they follow the same binding rules as with DNA, except that in the place of thymine, this is not to form a new double helix, but rather to give RNA the ability to read information from DNA. Now, tRNA reads information from the DNA and, in eukaryotes, transfers it out of the nucleus to an organelle called a ribosome. At the ribosome, mRNA gathers amino acids according to the information delivered by the tRNA. The ribosome is an organelle constructed primarily of rRNA. In it, the information on the tRNA can be read, and the amino acids gathered by the mRNA can be assembled into what we call a polypeptide chain. So a polypeptide chain is a precisely ordered string of anywhere from 80 to a couple million amino acids. The polypeptide chain is then transported to one of several possible places, including another organelle called a Golgi body. In these locations, the polypeptide chain is folded into an actual protein. Now, the shape of a protein is critical to its function. Therefore, electrical charges of polypeptides along the chain are critical to how the protein will form. Charged molecules will avoid other molecules with the same charge. For example, positively charged amino acid molecules will avoid other positively charged molecules, but they will seek out and attach to negatively charged amino acid molecules. If all goes well, which it doesn't always, a positive charge will find the exact negative charge to which it was intended to attach, and thus the polypeptide chain folds into a functional protein. To me, it's quite amazing how the charges are distributed along the polypeptide chain in the precise sequence necessary to shape the protein. We'll want to note that somewhere around 20,000 proteins are described in human DNA, although some of these might be altered after the polypeptide chain is produced, such that a single gene can generate multiple proteins. As a result, there are perhaps 1 million unique proteins in the human body, each performing some kind of specialized function. I want to just say one thing here, and that is something I don't know for sure, is whether these charges represent polarizations of the amino acids or they're actual charges on the amino acids. But one way or another, positive finds negative, negative finds positive, and the protein folds into its correct form. One thing that we will be considering soon is how important is the shape of a protein to its function. And so what occurs to me here is, how is it that the genome is able to arrange the amino acid molecules in so precise a way that the charges along the length will facilitate each precise folding and shaping? In thinking about this, I consider the range in the numbers of amino acids required to form the many proteins. Did this happen by chance? I don't see it. But this folding process can go wrong, and some diseases result from improper folding of polypeptide chains. Also, temperature can very much affect the shape of a protein and how it functions. Therefore, regulation of body temperature is vitally important in many organisms. Now that we've discussed the role of shape in proteins, I want to consider the role of that shape in how energy is processed in the cell. In fact, we should look at the entirety of energy processing, albeit from a high level. And the basics haven't changed much since your baby boomer 1960s biology. Plants, we'll call them producers, receive energy from the sun in the form of photons of visible light. The photons are absorbed by any of several pigments the most familiar being chlorophyll. Through the very complex process of photosynthesis, the plant produces carbohydrates and oxygen. This process occurs in organelles we call chloroplasts. Different pigments use different energy levels of light photons, which we see as different colors. The colors of light the chloroplast don't use bounces off, and that's the color we see. Chlorophyll gets energy from blue and red light, so it rejects green light. So you see green leaves. But for example, plum trees use a different pigment, and its leaves are a different color. But regardless of the pigment, the photosynthesis process produces carbohydrates which can convey energy if consumed by a consumer. And so what we call consumers, like us, take in oxygen and eat the carbohydrates. We burn the carbohydrates. As a consequence, we breathe out CO2 and produce water. But how does that actually happen? Let me assure you that it is really, really complicated. My description will only outline the events going on in your consuming body. Now, when I was young, I was taught that we are burning food in our bodies. That made some sense to me because my body was warm. But things that burn are really hot, and my body wasn't that hot. As a child and later in life, I puzzled over many possible ways that could be, but I never figured it out on my own. So we need to talk about how this happens. Spoiler alert, the processing of energy requires reactions to be catalyzed by those special proteins called enzymes. That's the key to the mystery. And this is why I didn't burn up when I ate my peanut butter sandwich. But it's a much bigger picture than that simple fact. Energy processing is really, really complex. And as you can see, it involves a symbiosis between the producers and the consumers. Let's start by talking about what happens when you set fire to a piece of paper. When we measure the temperature of a substance, either with our finger or a thermometer, we are measuring the average kinetic energy of the atoms and molecules. This might be a function of their movement around in a liquid or a gas, or it might be how much they vibrate while being held in place in a solid. When we heat something up, meaning we add thermal energy, as with a lit match, atoms and molecules vibrate faster and can be broken off of a solid. Now, in a liquid or a gas, these particles speed up, which means we'll say they're hotter. Of interest to us is what happens with atoms and molecules that will react chemically. For example, how it is that carbon atoms react with oxygen to form CO2, and how it is that oxygen and hydrogen react to form water. And not only that, how is it that we might get loose atoms of oxygen, hydrogen, and carbon to engage in these reactions? Well, paper is made of cellulose, which is a carbohydrate that happens not to be soluble in water. As you can see from its name, carbohydrates contain carbon and hydrogen atoms. So if you apply enough energy, as with your match, you should be able to shake carbon and hydrogen atoms loose. And assuming there is oxygen present, you should be able to get the resulting loose atoms to combine with the oxygen to form CO2 and water. But we see that hydrogen and carbon can both coexist comfortably with oxygen at room temperature and not react. Of course, when they do react, as in burning, they release energy. But what does it take to shake the atoms loose, and what does it take to get the released atoms to react with oxygen? Basically, the energy from the match will shake them loose. And then they'll have enough kinetic energy to crash into one another with sufficient energy to cause the reactions to occur. We call the minimum energy required to permit these reactions the activation energy, and we measure it as temperature. In the case of a match lighting paper, that carbohydrate we call cellulose, that's a temperature of 451 degrees Fahrenheit. That's a lot hotter than your body. Hopefully you remember from high school chemistry that a catalyst is a substance that participates in a chemical reaction, but when the reaction is over, it's unchanged from its original state. Here we need to catalyze these reactions so that they can occur at much lower temperatures. Enter enzymes, biochemical catalysts. As we said earlier, enzymes are proteins, and their shape is critical to their function, and not just their shape, also the locations of charges on their surface. In the case of enzymes, a critical feature of their shape is that they have cavities with charges lining them. So consider the interaction of an enzyme with a carbohydrate molecule. We'll call the carbohydrate the substrate, and we'll call the enzyme the catalyst. Long story short, the charges in precisely shaped cavities in the enzyme allow it to attach to the carbohydrate molecule in a way which allows it to tear the molecule apart. The protein tears the molecule apart by first engaging the carbohydrate molecule and then changing its own shape. This does require energy, but not nearly as much as the activation energy a match brings to a piece of paper. And we get more than enough energy back when we combine the carbons and hydrogens with oxygen atoms to make water and CO2. Now let's recognize something important here. The enzyme is shaped and contains charges exactly to fit the carbohydrates and other nutrient substances. And what of the loose hydrogen and carbons the enzymes release and the loose oxygens available in the cell? They need to combine to form water and CO2, but they also have an activation energy barrier. Energy is required to force the atoms together to cause them to react chemically. But from the standpoint of the thermal activation energy required to cause these reactions, this is much lower. So the temperature is much lower. Again, enzymes catalyze these reactions. Enzymes can capture carbon and oxygen atoms or capture hydrogen and oxygen atoms and force them together with enough force to form water and CO2. This takes energy, but more than enough energy is then released from these reactions to compensate for the loss. This warms your body up to 98.6. But, and this is a big but, how this energy gets fed back into the whole system in a useful form is really, really complicated. Which brings us to the topic of cellular respiration. Perhaps you've heard of the mitochondria as the powerhouse of the cell. For the most part, cellular respiration occurs in the mitochondria of eukaryotes and elsewhere in prokaryotes. Catalyzed reactions here participate in this incredibly complex phenomenon. In it, cells break down fats, digested proteins, and carbohydrates, including the carbohydrate we call glucose. It forces an energy releasing reaction between carbon and oxygen and then uses the energy to create molecules of a chemical called adenosine triphosphate. We've met this chemical before. It's ATP, and we think of ATP as the energy currency of life. In fact, when we taught cellular respiration in the classroom, we set up stations in a circle around the room for banks and stores, with students using a paper currency labeled ATP to make purchases and save their money. It's difficult to overstate the complexity of cellular respiration. A process called glycolysis acts on a fat or carbohydrate to crank out several chemicals. These feed what's called the Krebs cycle, which generates energy-containing molecules, including ATP, but it also produces the CO2 you breathe out. While the whole of cellular respiration is complex, the Krebs cycle is especially remarkable in its complexity. I really can't see how all of this could have come about in the incremental process demanded by the evolutionary process as currently articulated, mutations of the genome acted on by natural selection. There would have been a myriad of progressive steps involving new proteins and novel mechanisms, each in some way functional to promote the survival of the organism each step of the way. Has there been enough time and opportunity for random chance to work some kind of magic here? And a question we will come back to, is it reasonable to believe that time and the laws of probability alone can produce increasingly complex proteins and biological processes at all, even just one single new functional protein? Now, we will not go into detail, but for the sake of completeness, I'll mention that there are two manifestations of the Krebs cycle, one aerobic and the other referred to as fermentation. Your cells might shift to fermentation when the availability of oxygen is low, such as when you use up the readily available oxygen in your blood during exercise. A byproduct of fermentation is lactic acid, and this was believed to cause muscle soreness after exercise, but this viewpoint has been recently questioned. For our purposes here, we'll recognize that the aerobic cycle produces more ATP molecules than fermentation, so your body will prefer it. The process of yielding ATP from the Krebs cycle involves what's called the electron transport chain. Among other things, this leg of cellular respiration combines hydrogen and oxygen to produce water. We won't get into all of that since the details are beyond the scope of what I want to discuss here. So how is this ATP used? An example would be at the cell membrane when those gatekeeper proteins need to pump gases and nutrients uphill against the concentration gradient. Each molecule pumped costs the cell one ATP molecule. Now, there's a lot more to how a living cell functions, and you can see that it's a bit complex. Which brings us to the question of how did we get here? How did life begin? And is it reasonable to believe that natural selection, acting on mutations in the genome, can produce increasingly complex forms of life? For example, in what we call the Cambrian Explosion, could successive changes in the genome have differentiated between organisms with 2, 4, 6, and 8 legs? And how did life begin in the first place? Since the validity of the Miller-Urey experiment has been brought into question, can we still identify a way in which the amino acids necessary to produce proteins could have formed? This gets some rather heated debate, a lot of it focused on the transient nature of the reactants necessary to produce the precursor molecules in a proposed system requiring millions of years. But these discussions go beyond me, so I can't comment on them. On the other hand, there are some other questions that I find easier to contemplate. The leading hypothesis on the origin of life is called the RNA world hypothesis. We need DNA and RNA for life, but the thought is that RNA appeared first. We remember those strings of nucleotides with 4 bases in RNA: adenine, cytosine, guanine, and uracil. After the nucleotides connected up, this hypothesis says the RNA was incorporated into a system that permitted replication and eventually the development of a membrane and other features of the cell. This included the emergence of the double helix of DNA. And somehow, the cells learned to associate the nucleotides on the DNA molecule with specific amino acids. The machinery required to read the DNA appeared along with the machinery to gather amino acids and manufacture proteins. Of course, the nucleotides must have begun organizing in very special ways to represent the very special target proteins. Now, no one believes that this hypothesis has been developed well enough to provide confidence that we have a clear understanding of the origin of life. And some of us believe that it never will. One of the biggest obstacles to making this hypothesis credible is the sheer unlikeliness that a random assembly of nucleotides can produce a single functional protein. Recall that there are 20 amino acids available to be incorporated into a protein. The simplest genes in the human body consist of strings of about 80 bases, A, C, G, T, in the exact right order, but that number can range up to 2.3 million bases in a single gene. Again, all in the correct order. And what's the probability of getting from these 20 amino acids the correct selection of bases, all assembled in the correct order for the simplest gene by a random, unguided process? You will need a pretty big number. Now, I didn't do the math on this myself, but as I understand it, you should start with the total number of subatomic particles in the Milky Way galaxy: protons, electrons, and neutrons, and then add 2 orders of magnitude. So would there be enough time available in the window we understand from natural history to assemble such a sequence by pure chance, assuming that the other machinery was available to take advantage of it? Remember, this is for just one relatively simple gene. According to people who have crunched the numbers, absolutely not. To me, this constitutes the falsification that Popper called for. You can make up your own mind, but I sure don't think this works. I don't see how this could happen without an intentional mind behind it. According to William of Ockham, we should be looking for the simplest explanation that fits the observed facts. To some, especially those who follow materialist philosophy, looking for a supernatural explanation leads away from the point Occam's concept was making. On the other hand, what can we objectively make of the evidence? Can random point mutations on the genome lead stepwise to a new biochemical process? Is it reasonable to believe that mutations can be somehow stored up until they spring forth as a new body plan? Does DNA even store this kind of information? In fact, your DNA doesn't code for you having 2 legs and 2 arms. That's controlled by gene regulatory networks, what we call epigenetic information. Gene regulatory networks don't mutate the way genes do, and they are really, really complicated. So, how did new or modified gene regulatory networks appear if they're not a consequence of changes in genes? Maybe I haven't looked hard enough, but I have yet to see a hypothesis that even attempts to answer this question. In 1995, Daniel Dennett published his book, Darwin's Dangerous Idea. I read it as soon as it came out. In it, Dennett described the theory of evolution as a universal acid capable of eating through and destroying many aspects of culture, including our ideas of meaning and morality. And even if Darwin is overturned, the damage has already been done. There's no going back. And maybe he's right. For me, the metaphor of Darwinism as a universal acid works powerfully, but in a different way. Let me explain. I had once believed that there was a logical reconciliation between the work of the Judeo Christian God and the findings of modern science. That, in fact, the conclusions of science regarding life were valid, and things the Bible said about creation could be taken somewhat metaphorically. But when I began to learn the elements of molecular biology and the explanations scientists propose for how we got here, I became very disillusioned. Along with public intellectuals like Anthony Flew, David Gelernter, and Thomas Nagel, I couldn't reconcile what scientists were saying with common sense. There's no way, absolutely no way, that you can get from the very complicated process of mitosis, asexual reproduction, to the process of meiosis, sexual reproduction, in a stepwise process of point mutations on the genome acted on by natural selection. Meiosis is orders of magnitude more complex. What lies in between? No one knows. We call this irreducible complexity. We can't find evidence for or even imagine the incremental steps necessary to produce a biological structure or process. I'll also say, but with less certitude, that it's extremely difficult to believe that, in the context of the natural laws we understand, time and chance can encode the sequence of nucleotide bases necessary to describe the design of even the simplest protein alongside the machinery necessary to translate this information into an actual protein. The machinery and processes are required for the generation of mutations, and yet mutations are required to create these complex systems. The machinery is, of course, largely composed of proteins. Which came first, the chicken or the egg? How is that supposed to work if you needed the machinery to first make the proteins, but you needed the proteins to build the machinery? And does natural selection, acting on point mutations on the genome, actually lead to increasingly complex forms of life? Different, okay, but more complex? I don't know. Certainly, the concept works for what we call microevolution. For example, it appears that many years ago, the mutation in a gene responsible for a certain blood protein changed the blood of people living in Africa. This gave some people a survival advantage in that they were much more resistant to malaria. People with the mutated gene were much more likely to survive, and so that changed gene became nearly ubiquitous among African people. We know this today as sickle cell anemia, but it's much less beneficial when malaria is no longer a feature of the environment. Outside of an environment characterized by malaria, the trait is not beneficial, but it's still here. Beneficial or not, this mutation did not produce something more complex. It actually removed or deactivated a protein that was otherwise beneficial in an environment free of malaria. The mutation made the blood cell less complex. In school, we taught about the peppered moth in Industrial Revolution England. As black soot covered the land, light-colored peppered moths lost their advantage of camouflage and were devoured by predators. Dark-colored moths now had the advantage and proliferated. As presented in school textbooks, this was the emergence of a new species, but it was nothing of the sort. As the pioneering horticulturist Luther Burbank discovered, environmental pressure can certainly favor one variety over another and result in the proliferation of a newly favored variety. but a variety is not a species. And we never told the students that after the environment of the English countryside was cleaned up, light-colored peppered moths proliferated once again. So where can we see a mutation that defies the laws of probability and increases complexity? I haven't seen anyone propose a specific one. They can be postulated, and then a sequence of theoretical mutations might lead to a new biological structure. But when you try to dig into the nuts and bolts of how this might actually happen, finding a way to get to the smallest increase in the complexity of a single protein becomes a daunting task. Some decades ago, Stephen Jay Gould proposed something he called punctuated equilibrium, in which the models for certain traits could be stored up in the genome and then released under environmental pressure. But he failed to find the processes necessary to make this work, and no one else has either. The devil is in the details. And so Gould and punctuated equilibrium both died. In the context of questions like mine and the uncertainty left by other efforts to explain evolution, Dennis Noble and James A. Shapiro established the Third Way Evolution Project, in which they are exploring ways of thinking about evolution that would replace the existing scientific framework. Noble and Shapiro would bring back a form of something called Lamarckian evolution, which was thought to have been superseded by Darwin. In Lamarckian evolution, organisms change in response to the environment in which they live. For example, giraffes grew long necks in order to reach the leaves on tall trees. I don't know much about what they're thinking, and I'm in no way promoting their work. The third way is considered a fringe movement, but it does represent a recognition that the current paradigm lacks explanatory power. Professor Brett Weinstein and his wife are two well-known evolutionary biologists who acknowledge that the idea of natural selection acting on point mutations on the genome will not account for the apparently independent emergence of complementary structures, such as hemoglobin in the blood alongside the heart and the circulatory system. It will also not account for the relative speed of morphological changes we seem to see in the fossil record, such as the apparent evolutionary change of a small mammal similar to a shrew to a modern bat. He does not at all reject Darwin, but he says that the Academy is so wedded to what we'll call neo-Darwinian molecular biology that it has failed to properly investigate the inconsistencies in the neo-Darwinian narrative. He says that the Academy has ceded ground to those investigating what's called intelligent design. As a materialist, he rejects creationism, but he says the intelligent design people are actually doing good scientific work. He says that he is developing a new hypothesis regarding how the models for novel traits can be stored in the genome. I'll leave a link in the show notes to an interview in which he discusses his ideas. I've never heard him mention Thomas Kuhn, but he uses Kuhn's language. I will say that unless Weinstein can clearly identify the machinery and processes by which these models can be developed and stored and expressed, his ideas will go the way of Gould's punctuated equilibrium. I'll note that Weinstein is a little cynical about the state of affairs in the Academy. This might be influenced by his unpleasant experience at Evergreen State College in Olympia, Washington in 2017. So I am disillusioned. I'm not disillusioned with scientific investigation or the scientific method, especially in view of the rigors introduced by William of Ockham, Karl Popper, and Thomas Kuhn. But they need to be applied honestly. I think it's a serious problem for the integrity of science when a paradigm is actively promoted by scientists in the face of contradictory scientific evidence. Evidence that, when tested following Popper's system, fails to provide explanatory power. By all reports, if you are in the academic world of biology and you suggest that the Darwinian paradigm may be questioned, you will be canceled. Only extraordinary thinkers can escape this, and even they will be tarred. So for me, Darwinian evolution is the universal acid that ate away at my confidence that scientists present consistently honest interpretations of our universe, our world, and our lives. Is this limited to neo-Darwinian biology? I wonder. The problem with the current evolutionary paradigm is that it's based fundamentally on materialism, and any evidence challenging materialism is dismissed out of hand. As Dennett noted, the Darwinian theory is both elegant and beautiful. It seems to explain so much, but for me, it has failed. Does that mean I want peer-reviewed papers on biology to conclude that a gap in the fossil record should be filled by a deity? Certainly not. Should scientists stop trying to understand the origin of life, even in the context of a materialistic framework? No, but I also think it's wrong to claim to reach a conclusion that is so readily dismantled by the laws of probability for the purpose of rescuing materialism. In what's called the Cambrian Explosion, the fossil record appears to show the sudden appearance of significantly more complex life forms, at least in terms of geologic suddenness. Paleontologists say they believe this occurred about 530 million years ago. This is interpreted primarily from two fossil beds, one in Canada and one in China. As assessed by paleontologists, most of the modern animal types and body plans appeared during this time, perhaps during a period of 10 million years or less. The problem is that, as even Charles Darwin knew, we're having a hard time finding forms of life that we can interpret as precursors to the forms that then appeared, forms that would have evolved stepwise to become, say, sponges, snakes, and fish. What's interesting to me is watching the evolution of how this event has been interpreted in the Wikipedia. I've watched this over the years and couldn't help noticing how this explanation has morphed, apparently to accommodate the apparent need for a reconciliation with materialism. At least based on my recollection, there has been certitude about an explanation for the Cambrian explosion, although this explanation has been a moving target. Fortunately, this has morphed into a more nuanced discussion today. But the explanations have always been, to me, speculative. For me, when I look at the design of the cell, all of the machinery of life, the challenges to the current evolutionary paradigm, I see the work of an engineer. Maybe that's partly because I was an engineer, but it's the work of a designing, purposeful mind. Bacteria are driven through their environment by a flexible tail, a flagellum. The flagellum is driven by a tiny DC electric motor connected through a universal joint, just like ones that humans design. The motor has an armature, just like ones that humans design. Not only is this the work of a mechanical and chemical engineer, but also of an electrical engineer. The engineer's work has come a long way and seems to be directed to an endpoint. And the engineer seems to have a lot in common with us, junior engineers. This engineer is a lot smarter than we are, and his goals and methods might confuse us. While miracles are prohibited in a universe ruled by material laws and constants, they are suddenly more plausible. Perhaps someone could actually rise from the dead. And maybe, just maybe, this engineer had a special purpose when he made humans, and he wants to communicate with them. This would certainly be in his own way, and in a way that might not make sense to us. And so, if you got this far in my presentation, I'm glad you had the patience to hear me out. I hope I didn't offend you, at least not too much, and I hope you have a great day.